Hey guys, I really appreciate you hanging in there. We're going to start our next material for our test beginning with Chapter 14, Mendel and the Gene Idea, even though you have already been working with genetics in your labs. Genetics is basically studying transmission of traits from parents to offspring, of course. And used to be that people believed in blending, where you had traits that came from both parents and they blended together. In other words, if you had a tall parent and a short parent, then all the offspring might be medium in height. Well, we believe now in the particulate theory, where the parents are passing on discrete units called genes. Of course, when Gregor Mendel did his work, we didn't know about DNA yet. We didn't know about genes, mitosis, meiosis, none of that. Mendel worked with garden peas. He was a monk. And he worked with peas, and thank goodness that he did, because, for example, if he had worked with gerbils or mice or something like that, his genetics would not have been nearly as dependable. But nonetheless, the peas were available in lots of different varieties with lots of characteristics and traits. They had a short generation time, so he could look at inheritance over different generations. Of course, they produce a large number of offspring, and you can control mating. He actually took a paintbrush and moved pollen from one flower to another. Sometimes he would cut the flowers off that he didn't want breeding. He might cover them up with paper. But he chose only to track characteristics that had two distinct forms. For example, tall versus short, green versus yellow seed, round versus wrinkled seed. And he began with what we call true breeding varieties. So they were homozygous and all their offspring were identical. So just remember the word hybridization. That's what he was doing, is when you mate two contrasting true breeding varieties. A little bit more background. When we speak of the parents, we call them the P generation. Their offspring are called the F1. The offspring of the F1 are called the F2. Mendel came up with what we call the law of segregation. And this is extremely important. He said the two alleles for a characteristic that can be inherited separate when gametes are being formed and they end up in different gametes. And we've already studied this. This is meiosis. But it's very, very important to remember that if you inherited tall from mom and short in height from dad, one gamete does not get tall and short. We're going to divide them. Mendel said that alleles, which of course are different forms of the same gene, give the variations in inherited characters. Each gene is at a specific place or locus on a specific chromosome. For each character, the organism inherits two alleles, one from each parent. So that means when fertilization occurs, the offspring is going to pick up one allele from mom and one allele from dad. The alleles might be identical, like big H, or little h, and we call that homozygous. That means same in the zygote. Or they might be different, and we like big H, and we call that heterozygous, different in the zygote. One allele is dominant, and what that means is it will always be expressed. We usually use a capital letter like big H to represent that. The other allele is what we call recessive. It's not expressed unless it is inherited from both parents. And we normally are going to use a lowercase letter like little h for the recessive trait. So, excuse me, I know that you have been working with your Punnett squares, which shows you all the possible combinations of the egg and sperm, the traits that they carry when fertilization occurs. Remember, homozygotes are true breeding, heterozygotes are not true breeding because they have a recessive allele. A few more definitions. A phenotype is a physical appearance where a genotype is a list of the genes. So for example, if I have big P, and it is purple, those big letters in the genotype, the big P, let me know that purple must be dominant. So that when I have big p as a genotype, I know that is purple. But when I have the two recessive alleles, little p, that is white. A test cross is when you breed an individual and you're not sure of its genotype. Now, how in the world would you not be sure? Well, remember, genotype is a list of genes. You cannot look at an organism unless it has the recessive trait and know what kind of genotype it has. So we're going to take the individual with an unknown genotype and cross it with a homozygous recessive individual, and that will allow us to determine the genotype. Have a look at figure 14.7 in your textbook and let me know if you have questions about that concept, okay? Now, the law of independent assortment, which Mendel also came up with, says each pair of alleles segregates or separates independently of any other pair of alleles during gamete formation. Now, this applies only to genes that are on different non-homologous chromosomes, okay? And that's very, very important because it says, just because I have blonde hair does not necessarily mean that I will automatically inherit blue eyes. A monohybrid is a cross studying one character at a time. You've been working on some of those. A dihybrid is a cross studying two characters at a time. One of the things I want you to remember, genes that are located near each other on the same chromosome do tend to be inherited together. Mendel's laws reflect what we call the rules of probability. The alleles of one gene segregate or divide into gametes independently of another gene's alleles. We're going to talk about probability in monohybrid crosses. We're going to start with the multiplication rule, which states it's the probability that two or more, that plus means or more, independent events will occur together is the product of their individual probabilities. Have a look at figure 14.9. Make sure that makes sense. The addition rule says the probability that any one of two or more mutually exclusive events will occur is calculated by adding their individual probabilities. A multi-character cross is the same as two or more independent monohybrid crosses occurring simultaneously. To calculate the chances for the various genotypes, each character is considered separately, and then their individual probabilities will be multiplied. And we'll be applying some of these later. So you have been doing problems that are fairly straightforward, with a dominant and recessive trait, but inheritance patterns can often be more complex, and many heritable characters are not determined by only one gene with two alleles. So we're going to take a look at some of the exceptions to Mendelian genetics. Now, one thing I'd love for you to do is to go to my channel, Garrett, on YouTube, and I have worked a sample of all of these types of problems. And so maybe that'll be helpful for you. Remember, what we have been working with is called complete dominance, where the phenotypes, that means physical appearance, right, of the heterozygote, like big H, little h, and the dominant homozygote are identical. And so all that's saying is what we've been talking about, that big H, if it is long hair, will have the exact same appearance as big H, little h, because the big H, long hair, will be dominant. Incomplete dominance is an exception to the rule that says the phenotype of F1 is going to be between the phenotypes of the two parents. Now, one of the things I want you to remember about incompletely dominant problems as you're working them, they will always have three phenotypes. For example, if you are breeding snapdragons and you have red, which are homozygous, white, which are homozygous, and if you breed them and end up with heterozygous children, they will be pink. And that's not blending, it's incomplete dominance. Codominance is when two dominant alleles affect the phenotype in separate, distinguishable ways. And this is very interesting. You'll see an example of that when you work blood type problems, where you have AB blood type, where A and B are both fully expressed in the new individual. Now, one of the things to remember about dominant alleles, they're not necessarily more common in the population than recessive. So it's not a matter of, are there more of them? So, for example, 1 out of 400 children is born with polydactyly, which means you have extra fingers or toes. Guess what? That's caused by a dominant allele. But most of us don't have polydactyly. When we talk about the concept of multiple alleles, most genes actually exist in populations in more than 2 allelic forms. And a great example of that is blood type. Okay? So we know that we have blood type A and B and AB and O. Let's talk about that. There's an enzyme that is encoded by the big IA allele, which adds A carbohydrates to the surface of the red blood cells. That's how you get blood type A. There's an enzyme encoded by big IB allele, which adds the B carbohydrate to the red blood cells. That's how you get blood type B. There is an enzyme encoded by the little I allele, and it does not add either one of A or B, and that's how you get blood type O. Check out figure 1411 and let me know if you have questions about blood type. Another very strange, interesting concept is pleiotropy. Most genes have these genes that are, this is strictly pleiotropy that we're talking about now. These have multiple phenotypic effects. For example, if you inherit the genes for cystic fibrosis, and separately, another example is sickle cell anemia disease, then those genes will have multiple effects on the phenotype of the individual. We'll talk more about that later. Some traits may be determined by 2 or more genes. Epistasis means one gene affects the phenotype of another due to interacting gene products. For example, if you look at the coat color of Labrador retrievers, one gene controls the pigment, the color pigment. Another gene actually controls whether or not that pigment gets deposited in the hair. That's really cool, isn't it? So look at figure 14.12 and have a look at that. It's a very, very neat inheritance called epistasis. Poly means many, of course. Polygenic inheritance means multiple genes independently affect a single trait, and usually they're additive. For example, there are 180 genes that actually determine your height. There are a lot of genes that control skin color. Intelligence is another example. Check out figure 14.13. Let me know if you have questions about that. The environment can also have a big impact on physical appearance, the phenotype. The traits that depend on multiple genes often combine with environmental influences, and this is called multifactorial. What's really interesting, for example, is we know hydrangeas are a flower and they can be blue or pink. What's really interesting, the expression of that flower color is determined by the pH of the soil. If the plant is growing in a basic soil, the flowers are blue. If it grows in an acidic soil, the flowers are pink. The organism's phenotype reflects its overall genotype and its very unique environmental history. Human traits are Mendelian, but humans are not good for genetic research for a number of reasons, and let's talk about those. First of all, our generation time is too long. My goodness, by the time you have children, grandchildren, great-grandchildren, the researcher would be dead. The parents usually produce too few offspring, where a pea plant might produce thousands and thousands of children. Unfortunately, the average human isn't going to go much above 20, if that. And breeding experiments, particularly those that are done at random to answer research questions, really are unacceptable because there are so many ethical concerns. Now, one of the things that you've also been working with in lab is pedigrees. In other words, making a family tree, and it's used to describe generational relationships. It can also be used to make predictions about your offspring. With recessively inherited disorders, now it's recessive, right? Then the individuals have to be homozygous. Otherwise, the recessive would not show, right? Some individuals can be what we call carriers, and these are heterozygous individuals that carry the recessive allele, but their phenotype is normal. Albinism is a great example of that, which is lack of pigmentation in the skin and hair. Take a look at figure 14.16 and make sure you understand this concept. The risk of inheriting the condition is increased by consanguineous matings. That means between very close relatives. And we know that most societies have either official laws or certainly societal taboos against sex with relatives that are too closely related. Cystic fibrosis is the most common lethal genetic disease in the United States. Another one that we will be talking about and have been talking about is sickle cell disease. These are both inherited disorders. Some human disorders are caused by dominant alleles. If they cause a lethal mutation They're very rare. In other words, if the mutation causes a genetic change that kills the individual, those disorders are rare. One example of a very rare dominant allele is achondroplasia, which is a form of dwarfism. Have a look at figure 1418. Don't forget also the timing of the onset of a condition also affects its inheritance, like Huntington's disease. Huntington's disease, typically in the past when we had no tests for it, people had already married and had children before they ever knew they were afflicted with the condition. Multifactorial disorders include things like heart disease, cancer, alcoholism, many types of mental illnesses, and this is where we see both genetic and environmental components. And we know lifestyle can have a tremendous effect on phenotype, the physical appearance. Genetic counselors are very important because they study the genetics of the family, create pedigrees, and use the probability rules, trying to help identify carriers and define what are the odds that your children will inherit a certain condition. So please make sure that you understand figure 1419, because one of the most important things a genetic counselor can do is make sure that the couple fully understand the genetic testing that has been done and how those results are going to be used. There are several types of fetal testing. Amniocentesis, where cells are actually removed with a small amount of amniotic fluid. Chorionic villus sampling, and finally, ultrasound. We also perform newborn screening. Some tests now are routinely performed. For example, phenylketonuria. Please let me know if you have questions about any of the concepts in this chapter.